Dear students, good morning to all. In the session 54, we will be dealing with the respiratory and nervous system in freshwater mussel. Freshwater mussel is a molluscan and we have studied the externals and digestive system of freshwater mussel in the previous session. It is found in paddy fields, river bank, and it is a bivalve shelled animal with the wedge-shaped foot and a mental membrane. Within the mental cavity, we find a mass-like structure which contains the visceral mass. And if in this session, the learning objective are to study the respiratory system in freshwater mussel and to know about the nervous system in freshwater mussel. Coming to respiratory system, our respiratory system is gills or tinidia. There are two tinidia on either side of the visceral mass attached to the mental cavity. Mental membrane also works as an accessory respiratory structure. Exchange of gases occurs between water and the mental membrane. Tinidia are the chief respiratory structures. Two gills or tinidia are located in the mental cavity attached to the mental membrane, and they are like thin fold of skins protected by the mantle membrane. If we observe the structure of gill on either side, it consists of series of lamellate membrane-like folds which are known as gill plates or lamellibranch type of gill. Branchium or tinidium which contain the plates. Plate means sheet like membrane folds which are arranged on either side of the foot in the form of a W-shaped structure. The attachment of two gill plates makes it a bipectinate structure and inverted W-shaped structure. Each gill is contains two gill laminae. One lamina is inner and the other is outer. Inner and outer lamina are found on outer side and inner side of the mental cavity. Each lamina is V-shaped. The lamina is formed of two vertical plates called lamellae, which are inner and outer. So inner lamellae and outer lamellae form the two halves of the W structure of the gill, and lamellae are fused anteriorly and free dorsally. So free ventrally, and they are like folds of skin extending vertically from the mantle cavity, or they are lying on the lying on the adjacent side of the visceral mass attached to the mantle cavity. Each lamellae possess gill filaments. Gill filaments are structures which are found in the gills, and gill filaments are covered by cilia. The gill filaments are joined horizontally by interlamellar junctions. The lamellae of laminae are joined also are joined by interlamellar junctions. Filaments are joined by interfilamentar horizontal bars called interfilamentar junctions. So filaments are joined, and within the filament there are lamellae which are also joined. If we cut open the lamellae, in the junction we find there are vertical tubes or chambers which are known as water tubes. All the water tubes open into a large cavity on above this visceral mass and making the suprabranchial chamber. There is a communication from the chamber to the exterior, which is known as exhalant siphon. And inhalant siphon enters the water inside. Exhalant life siphon sends the current of water outside. And here is a cross section or vertical section in the region of foot. The visceral mass is cut in the region of foot, and we can see the W inverted W shaped two gills on either side of the foot. Each gill has a outer lamella, inner lamella, and outer lamella and inner lamella. Each of these filaments are again divided, and it leaves the suprabranchial chamber and infrabranchial chamber or mantle cavity. Mantle folds are seen on either side. So foot and mantle fold leave the W-shaped gill lamellae on either side, and each one having folds of skin with ciliated, again multiple times folded lamellae. So lamellae make the laminae, two laminae make the entire tenidium. There are two such structures which are arranged on either side of the foot lying in the mantle cavity. And next we have, see here, the section of the what? Gill lamellae. So there are two gill filaments. Outer gill and inner gill have two filaments each. The subfilamentar tissue is observed, and there are minute openings leading to the water tubes, and these are known as ostia, which allow water to enter into this and bathe these gill lamellae. Water current is produced by cilia on these folds of skin, which are known as filaments or lamellae. Filaments contain lamellae. Lamellae are joined by interlamellar junctions, and water tubes are the gap between two interlamellar junctions. So ostia open to exterior into the mantle cavity or the infra and suprabranchial cavity, which is directed to the inhalant and exhalant siphons. So each of these gill lamellae have ostia, water tube, interlamellar junction, and the folds of skin leaving the having the cilia, which are continuously beat and produce water current. Along with the water current, the food particles are also filtered by these cilia, which are found on the gill, and directed to the anterior region where the food balls are formed and swallowed. So feeding is also done by these cilia, which is known as filter feeding mechanism. So the portion of mantle cavity lying below the gill is called infrabranchial chamber. It communicates to the exterior via inhalant siphon. The lamellae are perforated by ostia located between the gill filaments and interfilamentar junctions. Through the ostia, water tubes open into the mantle cavity. So now how the mechanism respiration occurs. The beating of cilia of gills and mantle maintain a continuous water current into the infrabranchial chamber through the inhalant siphon. From here it enters into the water tubes, then to the suprabranchial chamber, then the water passes out through the exhalant siphon. Exchange of gases occurs when the water passes over the mantle, which is profusely circular, vascular, and carbon dioxide is expelled out to the outgoing water current. And coming to the nervous system in this animal, it is very much reduced and found in the form of ganglia and connectives and small nerves. There are three pairs of ganglia, anteriorly the cerebropleural ganglia and the middle visceral ganglia, and posteriorly in the foot region the pedal ganglia. All are bilobed structures. Cerebropleural ganglia lies above the mouth and innervates to the mantle, labial pulp, and anterior abductor muscle. Two ganglia of both sides of this paired ganglia are connected by commissures, which are known as cerebral commissures. Pedal ganglia are found in the foot region. They are also paired structures provided with connectives and commissures. They give nerves to the muscle of foot and to the statocyst. They are connected with the cerebral ganglia by a pair of cerebropedal connectives. Visceral ganglia are found in the region of visceral mass situated mid-ventrally on the abductor muscle. They are connected with cerebral ganglia by a pair of cerebrovisceral connectives. Sense organs are poorly developed in these animals. Freshwater mussels, they have statocyst and ostracium, which serve to check the water quality. Here we have a dissected portion of nervous system of this freshwater mussel. Here in the anterior region, there is a pair of ganglia, paired ganglia, which is connected by commissures to both the lobes are connected by commissures and with the visceral ganglia and pedal ganglia by connectives, visceroplural and cerebropleural and cerebrovisceral, cerebropedal connective and visceral ganglia, apparent structures and pedal ganglia in the foot. All the three are connected, and this kind of nervous system is primitive, which is found in this freshwater muscle. In the end of the session, we have now come to know what is the respiratory system present in freshwater mussel and mechanism of respiration, and also the nervous system that is seen in the freshwater mussel. Here at the end of the session, we have few MCQs which we can answer. The water enters the gill through the ostia, and the respiratory structure in freshwater mussel is ctenidia. The pedal ganglia is found in the foot of freshwater mussel. The sense organ seen in freshwater mussel is osphradium. The gap between lamella are known as water tubes. So we have certain reference textbooks. You can also refer the study material provided and answer the MCQs and get through the syllabus and write the